Assalamu alaikum guys. Okay, so this is Dr. Shah Fahad on your screen. I hope you're doing well. Okay, so today I plan on doing just a second. Okay, guys, so let's get started then. Bismillahirrahmanirrahim. Okay, guys, so biochemistry is actually a topic that a lot of the MBBS students find very difficult. So I plan on uh, making this a bit easy for you guys to handle it through your first year, second year, third year of MBBS studies. Okay, let's get started. The first topic that we will be doing here is amino acids. Okay, Bismillahirrahmanirrahim. Okay, guys, so what are amino acids? Any guesses? Leave it in your comment section. Amino acids are the building blocks of proteins, okay, as the name suggests, amino and acids. These are basically groups of organic compounds or biochemical compounds that contain both amino group as well as the acid group inside of them. Okay, amino group refers to the NH2 group, whereas the acid part refers to the carboxylic acid group. Wa alaikum assalam. Okay, someone has commented. Okay, guys, so let's just talk about amino acids in a bit more detail now. Let's look at their structure in detail quickly, after which we can move on. No, guys, you must have studied the structure already, by the way. Amino acid contains a centrally located carbon atom with a COOH group on the right side, NH2 group on the left hand side, hydrogen, and an R group. This R group represents the side chain of the amino acid, and it determines the type of amino acid as well. It can make amino acid acidic, it can make it basic, it can make it polar or non-polar as well. Polar or non-polar, depending on the type of side chain that it contains. Okay, now this is the acid group that an amino acid contains. This is the amino group that an amino acid contains. Hence the term amino and acid is given to it. So it contains two groups basically. Now, hence we also call amino acid amphipathic. What does amphipathic means, guys? Amphipathic means, amphi means dual nature. Dual. Amphi means dual. So it contains a dual nature. Okay? Oh, my screen is not visible. Thank you for telling me. Just a second. I hope it's visible now. Alright guys, so let's move on. We've spoken about amino acids, we've spoken about the basic structures of amino acid. Now the centrally located carbon atom. You also need to understand what the centrally located carbon atom is called. Any guesses what it's called? It's called the alpha carbon. Okay? It's called the alpha carbon. And hence the term alpha amino acid is given to these amino acids. Let's move on to the next part of the textbook now. So we just saw the overview of amino acid and its side chains and all the side chains, all the different properties that a particular side chain can give to an amino acid depending on what type of a side chain it contains. Okay, there are multiple types of amino acids, about 300 different types, but our body requires only 20 different commonly used amino acids. By the way, they are all alpha amino acids. Okay, alpha amino acid refers to the amino acid that contains an alpha carbon. Okay, our body can only consume alpha amino acids, cannot consume other amino acids. uh, Out of the 20, we've got essential amino acids and non-essential amino acids. Different books classify them differently, by the way. Okay, now at physiological pH, what actually happens at physiological pH? Now, this is an important question. When this amino acid is in body solution, so what actually happens? In body solution, you know that the carboxylic acid group and the NH2 group, the acid has this property of donating protons, right? Acid by definition is any substance that can donate protons, whereas base is by definition any substance that can accept protons, right? So the acid in body solution is gonna deprotonize, meaning it's gonna give away its proton and it's gonna form the carboxylate part, whereas the amino group is going to accept a hydrogen and it's going to form NH3 positive. This ion that has been created has equal and opposite charges on both the ends, hence it does not have any overall charge yet, so that's why it's isoelectric in nature. This isoelectric ion is called a Zwitter ion. Okay, so Zwitter ion is basically the ionized form of amino acid in body solution. Okay, we've spoken about that as well. Let's move on. Okay, how are amino acids linked to each other? Amino acids can actually be linked together to form proteins, right? Because amino acids are the building blocks of proteins. Now, the reaction by which amino acids join together and form different blocks of proteins is called, what is it called, guys? It's called condensation polymerization. Okay, chemistry. It's just the same thing, chemistry. So basically what happens is that in condensation polymerization, two amino acids, if you have an amino acid right here with all four bonds, COH and H2, everything else copied as it is, and if you have another amino acid right over here. Now the amino part reacts with the carboxylate part, the carboxyl part, okay? And this reaction is such that the carboxyl part loses an OH and the amino part loses an H, and when that happens, both of them combine together to form water and they're lost. Okay? Whereas this carbon atom and this nitrogen atom, both of them join together to form a peptide bond. Okay, this is how the peptide bond looks like. Carbon double bonded oxygen is bonded to a nitrogen, which is bound to a hydrogen. Okay? Everything else follows as it is. This reaction is catalyzed by a very special enzyme. You know that every single biochemical reaction in the body is catalyzed by a particular enzyme, right? At a time. So this reaction is catalyzed by an enzyme which is known as peptidyl transferase. Okay? Peptidyl transferase. And now, even though it has only one peptide bond, this is a peptide bond, okay? A new bond that has been made is called a peptide bond. Even though it has one peptide bond, this entire molecule is called a dipeptide because it's made up of two amino acids. That's the reason why we call it a dipeptide, even though it contains only one peptide bond. Understood? Okay. After this, let's move on. As I previously said, amino acids can be classified based on the type of side chain that they contain. Now look at all the non-polar side chains right over here. Obviously you don't have to remember every single amino acid from here, but you just need to know the very common one. Like for example, glycine is the smallest amino acid. Its side chain is hydrogen only. Okay, so that's non-polar. Similarly, this is also non-polar, this is also non-polar, this is also non-polar. All hydrocarbons are non-polar, right? So these are all non-polar, they are not soluble in water. Okay, non-polar amino acids. They are important for stabilization of the cell membrane. Okay. Walaikum assalam. Then we've got acidic amino acids, acidic side chains basically. In acidic amino acids, you guys can see that there is a carboxylic acid group right here, a carboxylic acid group right over here. Okay, which makes them acidic. Okay, they have a pH of less than 7. Then basic side chains. Basic side chains are the ones in which you've got an amino group as the side chain. Okay, amino groups are the side chains. Okay, so why a polar and non-polar amino acids are important? Polar and non-polar amino acids are important because the polar amino acids dissolve in water. Non-polar ones can dissolve in between the within the membranes. Okay, because you know that phospholipid bilayer is a non-polar membrane. Okay, on the inside, all the non-polar chains are present right over here. So the non-polar amino acids can dissolve in this non-polar chain, making it stable for this protein to stay within the membrane. You guys know that within the cell membrane you've got proteins, right? You've got transmembrane proteins. So the transmembrane proteins have non-polar amino acids that are dissolved in the non-polar phospholipid bilayer molecule. So the non-polar proteins, they help stabilize them within the membrane structure. The type of bonding that they form over here with the non-polar structures is called hydrophobic interaction. All right. Okay, let's talk about proline a bit, as it's mentioned here. Proline differs from other amino acids because it contains a ring structure. Hence, it's called an amino acid instead. It's not just called an amino acid, it's called an amino acid because it contains a ring. And this ring is such that it's making the nitrogen secondary, the carbon secondary actually. So it's actually a secondary amino acid rather than being primary. Okay, now what is secondary? It has a secondary amino group. Secondary, we refer to an amino group secondary when the nitrogen atom at the same time is bound to two carbon atoms. In this case, this nitrogen atom is bound to two carbon atoms. That's why this is secondary. This is a secondary amino acid, and hence it's termed as amino acid rather than amino acid. Okay. Now, amino acids with uncharged polar side chains, we've already spoken about them, but uncharged polar side chain. Now, this is important. They are polar, but they're not charged. Okay. One of the first example is disulfide bond. This disulfide bond exists only when you've got cysteine amino acid. Cysteine as the amino acid because cysteine contains the SH side chain. SH groups, when two SH groups come side by side, they lose the hydrogens, and as they lose the hydrogens, a bond is formed between sulfur and sulfur, making it a disulfide bond or a disulfide bridge. This disulfide bridge is a very strong bond and it is very important in maintaining and stabilizing the three-dimensional structure of a protein. Okay. Albumin that functions as a transporter is also stabilized by disulfide bonds, by the way, and it's the strongest of all bonds except for peptide bonds. Okay, peptide bonds are stronger than disulfide bonds, but these are stronger than the hydrophobic interactions, than hydrogen bonds, ionic bonds, all of those bonds. Disulfide bridges are the strongest because they are strong covalent bonds. Okay. We've spoken about all of this stuff already, so you can just go through it, read it. Now, something important right here: how you're gonna name the amino acids. The name of an amino acid in almost all of them would include the first three letters. Okay, that's how the nomenclature is done. The first three letters, because a lot of amino acids have the same first or second letters. So first three letters are used. Let's just move on. Okay, acidic and basic properties of amino acids. Now this is something a bit that is quite important. Amino acids have a dual nature, as you guys know already. They have an acidic group and a basic group. Okay. Now because they contain both the groups, they can act as a buffer. Now what is a buffer? Buffer by definition is a solution that can resist changes to pH when small quantities of acids or bases are added to it. Okay, that's a buffer. So amino acids And some proteins as well, they help maintain the blood pH. So for example, if H positive ion concentration increases in the solution, the amino group is going to react with it. It's going to use up all the H positive and it's going to shift the equilibrium to the left hand side. If the H positive ion concentration decreases in solution, then the equilibrium is going to shift to the right hand side to bring the H positive ion concentration back to normal. So in short, if base increases, if OH negative ions increase in solution, the acid part is going to react. If the H positive ions increase in solution, the basic part is going to react. Because of which the pH will be kept roughly the same, kept constant. This is an equation which is used to calculate the pH. Now, what is Ka? If you guys remember, if you have a weak acid and it dissociates, why are we taking a weak acid? Because weak acids are the ones that are usually used as buffers. Okay? Now, when a weak acid dissociates in an aqueous solution, it produces H positive ions along with A negative ions, right? Ka is the ratio of the products over reactants. So H positive multiplied by A negative divided by HA. What's pKa? pKa is negative log of this. Negative log of Ka. If you need to calculate the pH, pH equals pKa plus log of salt over acid, log of A negative over HA. What is this equation for? This equation is called Henderson-Hasselbalch equation. This is written right here. This equation is important because it helps us calculate the pH at which the buffer is present. Okay, this is used to calculate the pH of the buffers. This is another example in which it is showing how amino acids are acting as buffers. You guys don't need to go through this. This is never tested, okay? At neutral pH, we already spoke about that, so no need to worry. Okay, bicarbonate can act as a buffer as well. Yeah, that's true. And this bicarbonate is actually one of the major buffers of our blood, maintaining the blood pH. Okay, let's see how it works. Just a second. Where is the bicarbonate formed, or where does the bicarbonate come from? The bicarbonate comes from carbon dioxide, which is dissolved in water. Okay? Carbon dioxide that is dissolved in the blood plasma, basically. Carbon dioxide is coming from the tissues. So when carbon dioxide dissolves in water, it's going to form carbonic acid. And then this carbonic acid can dissociate to form H positive ions along with bicarbonate ions. Okay? Now, when the acid increases, acidic quantity increases, equilibrium shifts to the left hand side because it's going to get rid of all the extra H positive ions in the solution. Whereas when the acid concentration decreases and we need more H plus ions in the solution, this equilibrium shifts to the right hand side. You can look at it this way, or the bicarbonate itself alone can also act as a buffer, meaning when the acid concentration increases, it's gonna give away the H positive ion, because of which the H positive concentration in the solution is gonna increase, and when the acid concentration decreases, the reverse occurs, and it takes away the H positive ions. So that's how bicarbonate acts as a buffer. Rest of it is just simple. We just finished off the first topic. Now these are some practice questions. Even though the answers are given right here, make sure not to read the answers. Okay, and try solving these questions on your own first. Okay guys, then that's it for today. Inshallah, see you soon with another lecture. Take care.